So I'm going to talk about a single molecule label-free microscopy technique that I have developed as a postdoc at Chalmers in Sweden in the group of Christopher Langhammer. And now I'm at the Institute of Physics of the Czech Academy of Sciences, where I very recently started my own group, which plans to develop this technique even further. So, it is very easy to answer many of these fundamental biological questions. You just look at the thing. That was Richard Feynman's suggestion how to tackle the key biological challenges at the time. And by the thing, he meant a single biomolecule. And that might sound a bit arrogant, but that is, of course, like taken out of the context. He just wanted to push the biologists, to push physicists to develop better microscopy techniques. And since then, of course, many things happen. Now we have like the whole portfolio of microscopy tools capable of single molecule resolution that help us to basically uncover the complexity of life at single molecule level. So everybody in this room knows how difficult it is to see a single biomolecule. It's difficult, right? But maybe more relevant question is like how much we actually interfere with the native state of the biomolecule. Why we just cannot like zoom in into a cell and observe the dynamics of biomolecules in real time. This, of course, sounds like a science fiction. But the reason is actually simple, because biomolecules are tiny. They are nanoscopic particles. So, and since the scattering efficiency scales with the 6th power of its size, also the scattering efficiency depends on the optical materials of the particle, or differences between the optical properties of the particle and the environment, which for biomolecule is nearly nothing. So when you shine a light on it, only a very small portion of the light is scattered. But moreover, the biomolecules are moving in the environment, and they are moving quite fast, right? They are constantly pushed by other molecules, which is described as Brownian motion. And such a molecule can travel by, like in a fraction of a millisecond, can travel distances that are actually bigger than the size of the diffraction emitted spot, which means that limits the collection time. So basically what I want to say is that to shoot a video of such a molecule is like shooting a video of a black cat in a black box. And the cat is constantly moving. And, you know, we all know that many times we're even not sure that it's there, right? So it's extremely difficult. So what made it possible is fluorescent microscopy. It's, of course, a very powerful method, but it has its drawbacks. For instance, labeling can lead to undesirable modifications, especially for very tiny molecules. And also, like, we see only predefined targets, which means that the unlabeled but potentially important ones remain unseen. So these drawbacks have been the driving force for development of so-called label-free optical methods, like methods like iSCAT or localized surface plasmon resonance actually achieved to visualize like single binding events or single molecules. But these methods like rely on biomolecules, or they achieved to image biomolecules that are bound to a surface. And for many biological processes, like attaching or anchoring the molecule to the surface is not like the native state. So again, anchoring a molecule to a surface can lead to undesirable modification. So electrons, of course, are much more convenient for imaging. It's much more powerful than light. And before the last presentation, I wanted to say that you know, observing the dynamics, it's not possible, but that was my ignorance. We saw that, and it was a great talk. But still, the biomolecules need to be fixed. They need to be cooled down to cryogenic temperatures or dehydrated. So we wanted to address this challenge and to image biomolecules in real time and in their as closely native state as possible. So we developed this method that we called a nanofluidic scattering microscopy that images individual biomolecules without any labels, freely moving in solution, using surprisingly visible light. And we like to say that these are the conditions that mimic a natural environment. So the key component of the method is a nanochannel or series of nanochannels, very similar to the nanochannels that we saw in the previous talk. But our nanochannels are inscribed in a glass matrix. And size of these nanochannels can be in the order of tens of nanometers or hundreds of nanometers. Basically, they have to be bigger than the molecule. So the molecule can flow inside and freely diffuse. So then we shine a light on these nanochannels and collect the scattered photons using dark field microscopy. The same dark field microscopy that has been around for more than 100 years. So these nanochannels are quite efficient scatterers. So in any dark field microscope, you can see a nanochannel as a bright line. So the magic and the trick that allows us to see otherwise invisible things like small, tiny biomolecules is that the biomolecule and the nanochannel scatter coherently, which means that the total amount of the scattered light is not just the scattered light from the molecule and from the nanochannel, but there is this insignificant portion of the interference, and thanks to the interference, actually the contrast coming from that interference is several orders of magnitude more profound or bigger than the scattering from the molecule. So then, to image the molecule, what you have to do, you just simply subtract your background, which is the image of the nanochannel, and then you can image your molecule, and you can see the molecule as a dark spot. And voila, if you do everything right, which is not always the case, but if you do everything right, then you can see a protein freely diffusing inside the nanofluidics. So this specifically is a protein that is called thyroglobulin. So this molecule was not modified in any way. It doesn't contain any label. It's in room temperature, and the movement that we see is of course Brownian motion. Maybe I also have to add that the molecule actually diffuses or moves in all 3 dimensions. But what we see now, what might appear as a one-dimensional movement, is actually 3-dimensional movement. But since our nanofluidics is actually the size of the cross-section is smaller than the wavelength of the light, then the spot is actually much bigger than the nanofluidics. So this is another representation of the same video. Basically, it's a time-lapse image that we call hemograph, which basically shows the time evolution of the intensity profile along the nanochannel. And this dark, wiggly line corresponds to the trajectory of the protein inside the nanochannel. But we cannot only look at the dynamics or movement of a biomolecule inside a nanochannel. We can also observe or extract a lot of useful information. For instance, the optical contrast, or how dark this molecule, this spot is linearly proportional to the polarizability of a biomolecule, and it has been shown. For instance, by a method iSCAT, that the polarizability of molecule is linearly proportional to the molecule weight. So by measuring the optical contrast, we can measure the molecule weight on single molecule level, and at this moment with the precision of about 10 kilodaltons. But right now we're also working on a principle that will significantly lower this number. But what's important also to say that it will lower the number also for the whole range, for this whole dynamic range, which means that for bi-object in this megadalton regime, like for instance ribosomes, this kind of molecular resolution corresponds to a fraction of percentage of the total weight, which would be very difficult to achieve with conventional mass spectroscopy. So apart from the optical contrast, we can also get a lot of useful information from the diffusion, from the movement. We can track each individual molecule, and from the statistic of the movement, we can determine its diffusivity and determine its hydrodynamic radius. So together we have, from each molecule, we have two informations, the molecular mass and hydrodynamic radius. Okay, this is just a library of different proteins and short DNAs. So far, the smallest protein that is, a bit lost in a noise right now is BSA. That's several kilodaltons of molecule weight. So this is how a typical measurement of any sample look like. So each dot corresponds to each molecule that appeared in the sample. Different color corresponds to different molecule. There are 3 short DNAs, 4 proteins. What is interesting here that the protein actually, like the dependence of molecule weight and hydrodynamic radius follow certain line, and the DNA follows different line. But I want to say that that we were able to distinguish a molecule having the same molecular weight but different conformation. And that comes from the fact that DNA, because of the conformation, has very different diffusive properties than the globular protein. That might be hopefully interesting for future applications, for instance, for studying changes of conformation. It depends also on the time scales and etc. The proteins also have the populations of dimers. That's what we can see there. And specifically for ferritin molecules, there are some subpopulation that corresponds to molecules that have different content of iron in their core. Regarding instrumentation, the most essential part is the fluidic chip, which contains the series of nanochannels, which are connected to the sample containers that are connected to these nanochannels through these big microchannels. So, and the flow inside the chip we control by pressurizing the inlet. One notable advantage here is very low amount of the analyzed sample. Right now, a couple of microliters is enough to basically fill the whole system. And this is how the chip looks in reality. So it's like a centimeter square chip that is produced by a series of different nanofabrication, microfabrication methods. The nanochannels are inscribed by electron beam lithography. The microchannels are produced by optical lithography. And then at the end, everything is the the fluidics is enclosed by the glass lid using fusion bonding. And the fluidics is imaged by a dark field microscope that might not look like a traditional optical microscope. We built our own, and that's not because we like to play with the components. Actually, it's not only because we like to play with components. We are physicists, of course, we love to play with the components. But the other reason is that the conventional microscopes kind of lack the mechanical stability that we need, and it also lacks the efficiency of collection of the light scattering. So, yeah, there are some maybe improvements on the engineering side. Here I should also mention that with my colleagues from Chalmers University, we founded a spin-off company called NVU Technologies that is now turning this experimental platform into a commercial prototype. Okay, and apart from instrumentation, the absolutely fundamental cornerstone within this method is the effective and robust data processing. Unfortunately, I don't have time to talk about data processing so much, because I can talk about data processing for hours. It's like really my hobby. But I just like put here an example how it looks like if you would do exactly what I said at the beginning of the talk, that you extract the background of the image. Basically, you would see nothing. That's where we started. And the reason is that the background is quite busy image. So what you see is basically like the result of vibrations and drifts. So approximately like 2 years after that, or 1.5 years after that, when we developed like the proper data processing correcting for all the drifts and the vibrations, we were actually able to see the single protein diffusing. Like apart from what I would say it's like more traditional data processing, we also developed some like machine learning principles in collaboration with a group of Giovanni Volpe, where like of course like machine learning like beats the traditional one in terms of like computational time. I think the machine learning principles are like several orders of magnitude faster. So regarding applications, I have already mentioned the size and mass spectroscopy, but I believe there is potential for many more. Like right now, we are focusing on applying this method to investigation of biomolecular interaction. And just by like tracking of the individual biomolecules, I believe that we have like this unique tool where we can basically just watch the biomolecules bind and unbind at a single molecule level, freely diffusing without any label, basically without any like functionalization of the surfaces that is needed when you use like conventional techniques like SPR. So this is an example of chemograph that catches a binding event of 2 ferritin molecules creating a dimer. So we were actually like lucky ones. And I'm sure it's exciting, or for us it was exciting to see these like processes in real time. But from this one event, it doesn't really give you any like quantitative information, right? What you would need is much like longer time, so you would have the ability to measure the time scales of the bound and unbound states, and since these interactions are stochastic, you have to measure them many times. So then it's not so difficult to run like stochastic simulation of reaction and diffusion, and from that you get the information that for specific reactions with specific k-on and k-off parameters, you need to look at one molecule from like minutes to hours, which is of course much longer time than the molecule spends in the field of view of any microscope. So right now we are working on the idea to enclose like high number of molecules in something that we call nanofluidic reactor, which is basically just one long nanofluidic channel that has some means at the end to like enclose the molecules. We are working on two principles right now. One is based basically on like a passive enclosure of the molecules using like these constrictions at the end that basically creates electrostatic and entropic trap that keeps the molecule or prolong the time that the molecule spends in this volume significantly, or more like an active approach where we are developing this nanofluidic valve, which basically acts as any other valve in any other fluidic system. Like the idea here is to employ thermoresponsive polymers that are on plasmonic nanoparticle that could be switched on and switched on off optically through the plasmonic heating around the particle. And so far, I've been talking about proteins, but the imaging principle is not limited only to proteins. This is an example of another analyte that we successfully imaged, and that's lambda DNA. And in this case, the lambda DNA is not diffusing, but it's pushed through the nanochannel because of the applied pressure drop. And also, like the DNA inside the nanofluidic is stretched because of the confinement and because of the induced electrostatic forces. And in this way, we have also the opportunity to study like the time evolvement of the molecule density profile along the nanochannel. So we can see the evolvement in time. Here you might see like some parts of that that are darker. That's where because the DNA still have some kind of flexibility. So those are the portions of the time where when the DNA kind of bundles and stretches again. But I show this basically just an example for possible potential in the future to basically study like DNA and protein interaction where the protein would result in detectable change of the intensity along the DNA, and maybe in the future we could help to address the problem with the dark proteins that the fluorescents don't see. Yeah. It's also important to say that the configuration of this instrument enables not only label-free, but of course also fluorescent imaging format. And this dual modality allows us to directly compare between those two formats in the same instrument and exactly the same experimental conditions. So in this way, we would like to bring new insights into the ongoing debate about the impact of fluorescent dyes. So far, we have observed something that I guess everybody would expect. So it's nothing like groundbreaking, but if you like measure the length of the stretched DNA, the length of the labeled one is much. So these are longer compared to unlabeled one, which can be expected because the fluorescent dyes add some additional charge, which then results in a bigger stretch. So what is next? So 2.5 months ago, I've just started my own group in Prague, so it's very fresh. It's the group is called Dioscuri Center for Single Molecule Optics, and because it was supported in a Dioscuri program initiated by Max Planck Society. And the group will be focused on development of next-generation single molecule life science tools and their applications in life science. But in other words, I would like to give people from life science the opportunity to kind of dive into this nano universe and observe it in a way that was not possible before. But to be more specific, like our goal is to kind of make this nanofluidic scattering microscopy better, of course. We would like to achieve much better resolution. Like for instance, now we can see like these middle-sized proteins and large proteins, but we don't see these little guys, but that's the goal. Also, we would like to add some more functionalities into the method. Right now, I talk about the mass and size spectrometry, the interaction parameters and affinity, but we would like to put there also the measurement of the charge on a single molecule level or a characteristic fingerprint. In terms of applications, there are, of course, many ideas flying around. It's important to say that the partner of the center is Stefan Dietz from a technical university in Germany, where together we will develop applications regarding the molecule transport, where we will look at the basically molecular transport that is connected to microtubules. So to summarize my talk, nanofluidic scattering microscopy is a method that allows us to image freely moving single biomolecules in real time without any labels. It also provides for several quantitative information about their mass, size or conformation, interaction parameters and affinity. And I believe that this method presents a powerful complement to fluorescent microscopy in cases where labeling is unwanted or even impossible. And I hope it will find its applications not only in life science, but also maybe in the chemistry of colloids, surface phenomena catalysis, or mass transport and nanoscales. And we are always open for collaboration, like brainstorming with people who know who are from different fields of science. It's like, you know, the biggest. It's like the fun of my job. Yeah, I really love that a lot. So I would like to thank my previous colleagues from Chalmers University and Gothenburg University, and I would like to also thank the members of my team. We are not many. Some still haven't arrived, but they will. But you know, summer is long. And we are still looking for postdocs. I have right now like 2 postdocs position open. So if anybody would be interested, I would be very glad to talk about that. And of course, I would like to thank you for your attention.
Any questions? Oh, just time for one question. Please. One question? Mark, do you want to ask the one question?
I think it's a really amazing job you've done in getting this technique to work on single molecules. It's a real experimental tour de force. But there's something that I'm missing. Mm-hmm. If one wanted to determine the molecular weight or the diffusion radius, hydrodynamic radius of a molecule, one could use an ensemble of those molecules and do light scattering, and one wouldn't have to do as much averaging. So while this clearly demonstrates the ability to extract these numbers, illustrates the power of your techniques, it's obviously not the way to make those measurements when you have light scattering that doesn't require as much signal processing. So I'm sure there is something there, but what will you be able to see by looking at single molecules that you can't see by looking at an ensemble of molecules?
So I think the answer is probably similar to any like single molecule technique, that the power of looking at each individual molecule is that you can distinguish the heterogeneities in the sample. Because when you have like highly heterogeneous sample, everything is kind of smeared out, and you don't see, the subpopulations are very close. So I believe that like the real power of single molecules methods in general are actually in determining like the heterogeneities, and that might be in time, or like for instance for the interaction analysis, it might be heterogeneities in interaction, or heterogeneities really in size and and molecular weight.
I see your point.
Okay, so in the interest of time, we'll thank Barbora again.